Hello, and welcome to our final video on viruses. Um, this one won't be too long. This one is about viral load and viral growth, how we kind of cultivate viruses in the lab, how we can see them, and cytoplasmic effects on cells and how they can cause cancer. And finally, we'll differentiate once more between prions and viruses. All right, so um, a couple of things to note about the title here is viral load is the amount of detectable virus that we can detect. We'll get a short sequence of DNA and try to capture that through PCR or real-time PCR, and you can get a viral load. How many units of viral DNA can you find? And I did a little bit of this in graduate school, so the point about that is um, you can detect viral load uh, rather easily, but it's still subjective on what it actually means, because the viral load uh, doesn't mean you're not infected with a latent disease still. It just means that you cannot detect the limits. A interesting example of this is HIV. Um, Patients who have HIV can get their viral load down by using a cocktail of drugs or CART, combined antiretroviral therapy, or sometimes it's just ART, antiretroviral therapy. Um, so patients on CART or ART can actually lower their viral load to undetectable levels to where they don't pass it on to um, the unborn child or to a partner. Um, and so that is how that works. Um, but how that's related to this chart in front of us is almost exactly how it would go if you removed yourself from antiretroviral therapy. So while this is a graph that shows viral growth, the graph is eerily similar to um, coming on and then off of viral load. I'll explain. So just like we had our bacterial growth curve, we had our lag phase, our exponential phase, plateau phase, and then the death phase. Okay? In viruses, the legs of said graph are moved around a bit. At first, we go down, and then plateau, and then we go up, and then we stabilize. It's interesting that it's actually the exact reverse of the one up here. Just if we started here, worked our way down. So that is how a virus grows. The inoculum virus binds to cells, and then once they're in the cells, we can't detect it anymore. We can't detect levels of virion anymore. They're in the cells. So that's what I mean by viral load. Okay, and then once they release and quote unquote shed, sometimes you'll hear that bacteria shedding, they shed, and then now eventually they can come. They're gonna succumb to their host or reach a maximum threshold of growth. Viral cultivation and laboratory science. In order to produce the vaccine for the flu virus, back in the day, they would use eggs. They probably still do, but I think it's cheaper to make synthetic versions of it now. But they would essentially grow the grow the virus in the embryo of the chicken egg. And you can also make vaccines this way, or the flu vaccine. In our labs, though, we can see it this way, and we'll try to do these the last few weeks of class, where we grow phage. And you grow a lawn of bacteria, as you can see. The bacteria cover the whole plate, and then you put on a dilution of bacteria. The dilution is like really light here, a light dilution, so you can see those dots. And then over there, the dilution is probably tenfold higher, and you see tenfold more, and the same on the last plate. So it's like a negative stain in that you don't actually see the virus, you're just seeing that it's destroying the bacteria nearby it. So we move on to cytopathic effects. They are distinct observable cell abnormalities due to viral infection. And so lots of things can happen. You can lose your ability to adhere. You can change your shape from flat to round, shrinkage of cytoplasmic membrane, and just complete cell lysis. So eventually. So you'll want to know that these viruses can essentially cause cancer based on their cytoplastic effects, usually enlarging or performing a different function. And finally, we'll end with viroids and virusoids are types of other non-living infectious agents. I would just know those as distractors instead of picking like prophage or provirus. Prions, on the other hand, are important to remember. They were our first case study, and we'll look at them some more. So prions are proteinaceous infectious particles. They have their own life cycle, as we saw, and yeah, it could just be as simple as a misfolded protein. Well, that concludes our viral lectures. Hopefully we answered all the learning objectives for you. The phage therapy on the food was probably the best example of gene therapy that we will discuss.